हॅलो, ऐकताय ना सगळे व्यवस्थित? Then see the next slide. What is the outline of this topic? Outline means objective of this topic. तुम्हाला सगळ्यांना ही स्लाइड दिसते का? Outline of this topic. Hello. Hello. Give the answer from anyone student. कोणीतरी एकानं सांगा ऐका येते का नाही? Hello. Can I continue? Shall I continue? Okay. Then see the outline of this topic. First, aim of the DNA isolation. Why DNA isolation is important? Aim means to isolate the DNA from given blood or tissue. Second, what is the definition of the DNA? It is the segment of chromosome. This is the definition of the DNA. And this part is completed in your previous year standards, 10th, 11th, 12th, etc. Next is definition of DNA isolation. What is the definition of DNA isolation? How to isolate the DNA or extract the DNA. Next is what is the purpose of DNA isolation? Purpose means isolated DNA is used for the various applications. For example, PCR means polymerase chain reaction. It is used for the detection of disease. Recently, all of you know RT-PCR test for the corona detection. For that, DNA is very important. Also, other applications are there: detection of mutations, detection of criminals, fingerprinting, etc. Next is sample selection for the extraction of DNA. Which sample? Blood, tissue, virus, bacteria, etc. These samples are used for the isolation of the DNA. And last part is steps of the DNA isolation, means standard operating procedure of the DNA isolation. These steps are given in your journal, page number 187. That is the DNA isolation from blood and tissue. And last bit is chemistry behind it. All of you know this topic is completed. Nucleic acid chemistry, nucleoside, nucleotide, then ribose or deoxyribose, sugar, then purines, pyrimidines, structure of DNA, RNA. And these are the chemistry, and chemistry part is completed by Dr. Nilisha Werneker, madam. Then see the next slide. This slide shows the introduction of DNA, about the DNA. And this part is completed. Also, DNA is the macromolecule that carries genetic information from generation to generation. All of you know this point. And single mammalian fetal cells contain only few picograms of DNA. How many? Few means 10 raised to 12. Also, genetic engineering involves the manipulation of the genetic material. This part will be completed in the topic of recombinant DNA technology. Also, some part of this topic is completed in your 12th standard. For example, vector selection, primer selection, and there is modification is there in the genetic material. This process is called as genetic engineering. Also, almost all the experiments dealing with the gene manipulation requires pure form of the DNA. That's why isolation of the pure form of DNA, this process is very important for the genetic engineering process. And last point is, so isolation of DNA is important process is genetic engineering. Then see the next slide. Have you seen this slide? Structure of DNA. Hello. Structure of DNA. This part is completed. DNA is two standard double helical structure. One strand from 5' to 3' and another opposite direction. All DNA contain purine and pyrimidine bases, adenine, thymine, and cytosine and guanosine bases. Adenine bind with thymine with 2 hydrogen bond, and guanosine adenine, thymine, guanosine, cytosine by 3 bonds. That's why this bond is stronger than A to T bonds. Also, this DNA contain purine and pyrimidine bases. Also, all the purine and pyrimidine bases are held together by phosphodiester bonds. Also, this DNA contain deoxyribose sugar. And the structure of DNA means Watson and Crick model of DNA. This part is completed in the nucleic acid chemistry topic. That's why let's move to the next slide. Have you seen this slide? Anyone? Give the answer. DNA isolation method. The isolation method of the choice is dependent upon which method is selected. It depends on some parts. For example, what is the source of DNA? DNA is isolated from which material? For example, blood, then tissue, bacteria, and viruses, etc. These are the sources of DNA, and from these sources, DNA will be extracted. Then what are the applications of the DNA? For that application, which method is selected? For example, polymerase chain reaction means PCR. What is in the PCR? PCR means multiplication of the DNA. Then restriction of the DNA. It is used for the checking of some activities, mutations, etc. Then next part is DNA sequencing. What is the proper sequence of DNA? Will match by fathers or sons. Next part is fingerprinting. It is used for the identification of criminals. And last part is library construction of the DNA, etc. These are the applications of the DNA. And what are the types of DNA? All of you know types of DNA: genomic DNA and plasmid DNA. And to a lesser extent, the number of samples to be processed by robotic or automations. Huh? Also, these advanced techniques are used for the above applications. Then this slide shows the methods of isolating DNA. Are you listening to this slide? Homogenize. Methods of isolating the DNA. First, select the what is the tissue. For which tissue homogenization is required? For example, tissue homogenize chemically or mechanically. Homogenize means proper mixing of that proper tissue. It is called as homogenize. But for blood sample, there is no need of homogenize. It is required for the tissue purpose. How it is homogenize? Chemically or mechanically. Some chemicals are used or some grinding machine or homogenized machine is required here. Then two for the single cell suspension. After homogenization, we will get single cell suspension. If blood is used, there is single cell suspension is there. Then next part is cell wall rupture. For example, for the removal of RBCs and WBCs, the rupture of cell wall is very important. And for this rupture, some enzymes are used. Also for the rupture of some bacteria or yeast and fungi, some chemicals or enzymes are used. For the rupture of bacteria, lysozyme enzyme is used. And for the rupture of yeast and fungi, zymolase enzyme is used. After this rupture, then after centrifugation, this bacteria and yeast fungi will be floated. And after the removal of supernatant, this bacteria and yeast fungi will be removed. This is called as rupture of the cell wall of the bacteria and yeast or fungi. Then see the next part, cell membrane rupture. For this cell membrane rupture, some detergents are used. These are SDS. Long form of the SDS is sodium dodecyl sulfate or sodium lauryl sulfate. Also, sarcosine is used. Also, another chemical, Triton X100. This is another chemical used for the rupture of the cell membrane. Which cell is there? Bacteria, yeast, also RBCs or WBCs. Then also for this purpose, another enzymes are used. For example, proteinase. Here, proteinase K enzyme is used. Also, pronase E enzyme is used. Also, sometimes some chelators are used. There is one example of chelator is EDTA, means ethylene diamine tetraacetic acid. Also, there is one another enzyme, guanidine thiocyanate or chloride or urea is used for the rupture of the cell membrane. Okay, this is the rupture. Then see the next slide. How to isolate, means isolation of DNA by method. This is the short procedure of the DNA isolation method. Later on, we will discuss the detailed procedure as per your journal. First, see the what is cell extraction. How to extract the cell. For this extraction of cell, some chemicals are used. For example, phenol and CHCl3 means chloroform. Also, high salt of guanidine thiocyanate or sodium chloride or guanidine chloride is used. These chemicals are used for the extraction of the cell, means for example WBCs or RBCs. Next is removal of cell debris. Debris means after the degradation of the cells, some compounds are liberated. For example, cell contain proteins, lipids, and carbohydrates, means polysaccharides. For this removal of these compounds, this process is called as removal of cell debris. Also, for this, some chemicals are used. Means by these chemicals, RBCs, WBCs, and cell debris, means proteins, lipids, and polysaccharides are first removed. Then there is concentration of the DNA. For example, precipitation of the DNA. Some chemicals are used. For example, ethanol, isopropanol with salts. For example, sodium, lithium, and ammonia is used. Also, some another chemical, DNA absorbing matrix. Here, silica or silicon dioxide is used. Also, one cytidine, acetic boric acid. This chemical is used. Also, PEG means protein energy. This glutamate is used. Then also supermidine is used. Later on, we will discuss about the long form of CTAB and PEG. Also, supermide. These chemicals are used for the concentration of the DNA. Mostly here, alcohol or isopropanol is used for the precipitation of the DNA after the removal of all cells, RBC, WBCs, and carbohydrate, lipids, and proteins. Then there is one optional step is there, removal of RNA. Because if there is this process is not done, RNA is present in that precipitation. That's why RNase enzyme is used for the removal of RNA. And for every removal of the cells, first rupture the cells, then centrifugation and remove the supernatant. Then repeat the same steps by two to three times. Then completely removal of that cells. Also removal of RBCs. For RBC lysis buffer is used. For the removal of WBCs, WBC lysis buffer is. For the removal of cell debris, there is SDS, chloroform. These chemicals are used. And finally, RNase enzyme is used for the removal of RNA. After the addition of these chemicals and centrifugations, by the removal of discards, these compounds will be discarded and remaining compound present the DNA, and this DNA is precipitated by addition of alcohol or isopropanol. This is the short procedure of the DNA extraction. Then see the next slide. DNA extraction. How DNA extracted? Here, one tube is there, test tube. Mostly here, centrifuge plastic tube is used. Here, 15 ml centrifuge plastic tube is there. This is used for the important for the centrifugation process. First, in first steps, cells are lysed using a detergent that disturb the plasma membrane. First here RBC's membrane. Also, if there is RBC lysis buffer is not there, You can utilize pure distilled water. By distilled water, RBCs are ruptured, and after the centrifugation, WBCs and remaining parts are settled down at the bottom, and remaining solution contain RBCs. After the removal of that supernatant, RBCs will be removed, and this process is repeated by the two to three times. Then completely RBCs are removed. Then also WBC, yes, some other chemicals are used for the rupture of the WBCs. And by this procedure, similar as RBCs, WBCs are removed. Then carbohydrates, lipid, proteins. The next step is cell content are treated with protease to destroy the protein and RNAs to destroy the RNA. For this process used for the removal of proteins and RNA by centrifugation process. Third step, cell debris means carbohydrates, lipids, and protein. After the rupture of the cell membrane, these compounds are enter into that solution. For the removal of that cell debris solution, the supernatant containing DNA and transfer to clean tube. Here in this step, after centrifugation, cell debris will be settled down at the bottom, and supernatant contain the DNA. This DNA is transferred to the another tube, and in this dissolved DNA there is addition of ethanol or isopropanol. Then that DNA is precipitated, supercoiled or spooled DNA is formed. We will get DNA just like a thread, 10 to 20 millimeter thread. That thread is called as DNA, and this DNA is used for the application purpose. Then see the slides. DNA extraction methods. Some methods are used for the DNA isolation purpose. Which methods are used? For example, several common methods are used for the DNA isolation. First, see the organic extraction. Means here chemicals are used. Then what are the organic extraction method? There are some advantages and disadvantages are there. Here we are discuss only single advantage and disadvantage. In organic extraction method, we will get high quality DNA. High quality DNA. What is the use of high quality DNA? It is used for the genetic engineering purpose. This is the one advantage of organic isolation of the DNA method. Then there is one disadvantage is there. This method is toxic and time consuming because here guanidine thiocyanate, this chemical is used, cyanate or cyanate is used. That's why this method is toxic. Also, this method requires the long time. It is time consuming method. This is the disadvantage of the organic extraction method. Then let's move to other next method, Chilex method. It is called as chelation method. For this chelation, EDTA is used. Also, chloroform is used or urea is used. These chemicals are used. These are the chelating agents. Advantage is very fast and not toxic. This is the advantage of this method. DNA is isolated very fast, and this is non-toxic method. But there is disadvantage. Product will be impure. Here, isolated DNA will be impure. That's why this DNA is not used for the, not proper for the genetic engineering process. Also, not to be suitable for DNA sequencing, restriction, etc. That's why mostly this method is not used for the isolation of the DNA. Third one is spin column extraction. This is another method. It is automatic method. Here instruments and DNA isolation kits are available. This method. Advantage is fast isolation of DNA. It's high quality of the DNA and to be processed robotic or automation. This is the advantage of spin column is DNA isolation kit method. And there is one disadvantage, still toxic and technology consuming. This is the disadvantage. Sometimes it will be toxic, and here technology is used by this method. Then see this slide, organic method. Which chemicals are used for the organic method? Then first is the advantage of highly negative charge of the nucleic acid. First is lyse cells with sodium dodecyl sulfate, or PK means proteinase K, or DTT. SDS means it acts as a detergent for the removal of cell debris, carbohydrates, lipids, and proteins. Then PK, or this SDS is used for the solubilized cell membrane, RBCs or WBCs. Then next chemical is PK means proteinase K. Here this enzyme is used for the removal of proteins. Also RNase A is used for the removal of RNA. Then next is DTT. It acts as a reducing agent. Also, it breaks the disulfide bonds in strong proteins like proteins. These chemicals are used for the lyse cells with SDS, PK, and DTT. After the removal of all these compounds, add equal volume of the phenol. Then here, protein fragments and lipids are attracted to hydrophobic phenol, and nucleic acids are attracted to water. By this process, proteins and lipids are removed. Okay, this slide shows the organic extraction method. Here first, crude lysate containing nucleic acids and other cell constituents. Lysate means homogenization. For blood, there is no required. If tissue is there, lysis is required. Then mix thoroughly with equal volume of the organic solvent. For example, phenol, chlorophyll, and phenol chlorophyll. Then next stage is centrifuge. By centrifugation, 2 for aqueous phase contains water, soluble nucleus, soluble molecules, including nucleic acids. This aqueous phase contains water soluble molecules. That molecules contain nucleic acids. Also, that contain proteins and lipids become trapped in the organic phase. These are trapped and are thus separated away. Insoluble plant debris become trapped by interface between the two layers. Okay, and collect the aqueous phase. At the bottom of the tubes, we will get this cell debris or some RBCs and WBCs. Then collect the supernatant in another tube. That another tube contains dissolved DNA. By the addition of phenol or isopropanol, DNA will be precipitated. See the next slide. Add alcohol and salt to precipitate nucleic acid from the aqueous fraction. Here DNA will be precipitated. DNA are seen just like a thread like structure. एका छोट्याशा 10 ते 20 मिलीमीटरच्या दोऱ्यासारखं दिसतं ते. After centrifugation, there is supernatant and pellets are there. Pellets down nucleic acid. That pellets are washed with 70% ethanol to remove some other compounds or residual salts or other contamination, and discard ethanol to allow the pellet to dry. This process is very important for the formation of pure form of the DNA. Pellets are settled down. That pellets contain nucleic acid, and these pellets will be dissolved with water or TE. TE means buffer of tris and EDTA, and that solution contain pure form of the DNA. This DNA will be dissolved in water or tris EDTA. That DNA sample is used for the PCR purpose, DNA sequencing purpose, restriction purpose, fingerprinting purpose, etc. Okay. Here this organic method is completed. Then see the chemical methods. Here enzyme lysozymes are used. This enzyme breaks the peptidyl glycan of the cell wall. This rupture the structure of the cell wall. Also, proteinase K is broad spectrum protease enzyme. It digests protein. Also, inactive enzyme which can degrade DNA or RNA during purification. Also, these chemicals are used by chemical method. But recently, organic method is used for the DNA isolation or extraction method. Then see the next slide. Direct purification of the DNA. How to purify the DNA? And purified DNA is very important for the several applications. See the long form of CTAB, cetyl trimethyl ammonium bromide. This chemical is used to form the insoluble complex with DNA. This is for purification purpose. Cetyl trimethyl ammonium bromide forms insoluble complex with the DNA. After centrifugation, precipitate is formed, suspended in high salt solution. That high salt solution which is the nucleic acid. Also, there is silica gel particles from light binding with DNA. Light binding with DNA, it's small binding. In presence of guanidine gonidinocytonate column chromatography is run. Here, this chromatography technique is used for the purification purpose of the DNA, but recently this technique is not used because DNA isolation kits are available. Spin method is there. This is difficult method. That's why DNA isolation kit method is used. But also in this method, cyanide is used. That's why this method is toxic. But this method is used for the purification purpose of the DNA. Then see the slide. As per your journal, separation or precipitation of the DNA. Name of this method is phenol chloroform extraction method. This slide shows the material required for the precipitation of the DNA. First is blood sample or tissue sample. Second is RBC lysis buffer. Now in bracket 10 millimolar potassium bicarbonate, 0.15 molar NH4 ammonium chloride or 0.1 millimolar ethylene diamine tetraacetic acid. Also, instead of these chemicals, direct you can use the distilled water. This distilled water rupture the RBC membrane for the removal of RBCs. Next, 20% sodium dodetyl sulfate. This is used for the removal of WBCs. Next is proteinase K. This is used for the removal of proteins or degradation of disulfide bond. Also next is ethanol. And then trisphenol. Removal of cell debris, carbohydrates, lipids and protein. Then next is chlorophyll. Also isopropanol is required. These chemicals are used for the precipitation of the DNA. And last is tris EDTA buffer. It is used for the DNA dissolving purpose, or nucleus free water is used for the dissolving purpose. These chemicals are required for the DNA isolation method by phenol method, phenol extraction method. Then this slide shows the procedure of DNA isolation by phenol extraction method. This procedure is given in your journal at the page number 889. See this procedure. Add 3 volume of the RBC lysis buffer to 1 volume of the blood buffer in a tube and vortex vigorously for 5 minutes. Mix these chemicals for 5 minutes. Take blood and add 3 volumes of the blood of the lysis buffer or distilled water. Then mix by vigorously for 5 minutes. Also here inwardly mixing is okay. Then second, centrifuge the tube at 2000 rpm for 10 minutes. Centrifugation is there. After centrifugation, discard the supernatant. Then large amount of RBCs will be removed. Then repeat the RBC lysis steps for 2 to 3 times until while pellets is obtained. By this procedure, RBCs are completely removed. Then suspend the final pellets in 0.5 ml superintendent. In their superintendent process, clearly filtered, that is 0.5 ml. Resuspend this filter. Then add 60 microliter proteinase K solution. It is used for the removal of proteins or degradation of disulfide bond. And then add 20% sodium dodecyl sulfate and incubate at 56 degrees centigrade for 2 hours in water bath. By this process, proteins and some WBCs are ruptured. Then next slide, next step, add 1 ml of tris phenol solution to remove the above solution and mix for 1 minute. Add 1 ml of tris phenol in the above solution and mix for 1 minute. Then centrifuge at high speed, 13,000 rotation per minute for 10 minutes. Then collect the upper layer carefully and mix with 1.5 ml chlorophyll and mix by inverting 10 minutes. Means after the centrifugation, WBCs and the cell debris are settled down and clearly solution contain DNA. There's a settled down component contain WBCs. Cell debris means carbohydrates, lipids, and protein. These are settled down at the bottom and clearly solution contain DNA. This is transferred to the another tube. Also inversion by 10 minutes. Then again recentrifuge at 13,000 RPM for 10 minutes. Then here there is removal of completely cell debris, WBCs, proteins, carbohydrate, lipids, etc. Then collect the supernatant in a 1.5 ml tube containing 1 ml chilled alcohol. Gently swirl. DNA will be precipitated. That this supernatant will be transferred in the another tube. That tube contain chilled alcohol is there. Here ethanol or mostly isopropanol or propanol is used. And in that propanol, after vigorously shaking, DNA will be precipitated just like a thread-like structure, means dorasarka astate. Then centrifuge it at, again recentrifuge at 13,000 RPM for 10 minutes. Here recentrifugation is there. Then see the slides. Decant the supernatant, means discard the supernatant. Then wash with 1 ml 70% alcohol. In that bottom, add the 70% alcohol to remove the impurities. By this process, pure form of DNA will be isolated. Then mix the pellets and centrifuge it at 30,000 RPM for 10 minutes and again discard the solution. Here this is the used for the washing purpose of the DNA. Keep the tubes on a tissue paper in an inward position for 3 to 4 hours to air dry. By this process, complete solution will be removed. Complete alcohol is removed by this process. Then add 50 microliter tris EDTA buffer or nucleus free water and keep in water bath at 60 degrees centigrade for 1 hour. This is required for the dissolving purpose of the DNA and quantify using ultraviolet or visualize spectrophotometer at 260 nanometer. Visualize DNA by this ultraviolet rays or spectrophotometer. Then quality check can be done on the 1% agarose gel electrophoresis. After this lecture, Nilisha madam will take the another technique, electrophoresis. Okay. This slide shows the net non-phenol chloroform extraction method. This slide shows non-phenol. Other method is used. Here, plus in box of blood is there. First cell lysis is there. After centrifugation, transfer the clear in the another tube. Here, isopropanol or alcohols and salt precipitate nucleic acid from the aqueous solution. Then take the supernatant, remove the supernatant and pellets are there. Then addition of alcohol for the washing purpose and dissolved by DNA in tris EDTA buffer. It is the small procedure. Also, this slide shows the method of the lysis. This part is completed. Here, 2 to 3 times RBC lysis, 2 to 3 times WBCs or RNA lysis, 2 to 3 times protein lysis, and last is DNA precipitated. Then see this slide. As per your journal, DNA isolation from tissue. The tissue has to be homogenized by one instrument, homogenizer, or snap frozen in liquid nitrogen and mortar grinded in phosphate buffer saline. Here, phosphate buffer saline is used for the homogenization of the tissue. After homogenization, proceeds to steps 6 to follow all remaining procedure. In above procedure, proceed all steps from 6 to last as per above protocol for the DNA isolation from blood. Same procedure is there. Also, this slide shows the how to DNA store. DNA will store for 1, 2, 3, 4, 5 years. And this is required for the utilization of the DNA. For the DNA storage, some solvents are used. For example, DNA, RNA, and oligonucleotides are stored in 1x tris EDTA solution or 1 millimolar ethylene diamine tetraacetic acid solution or tris HCl solution. And pH of that solution will be between 7 to 8. In slightly alkaline pH is used for the DNA storage purpose. And which storage purpose is required proper for the DNA storage? For everyday use, plus 4 degrees centigrade is sufficient. Routine freeze temperature is sufficient for the everyday. But for the storage of long time, minus 20 to minus 80 degrees centigrade is required. Also, under 70% ethanol as a pellet, DNA or RNA can be stored at plus 4 degrees centigrade almost indefinitely. This temperature is required for the DNA storage purpose. Mostly everyday purpose 4 degree and for long purpose minus 20 to minus 80. Also this slide, see the slide. Okay, how to separate the DNA? Separation of nucleic acids. DNA molecules of different sizes can be separated by gel electrophoresis. This part will be completed after this lecture, electrophoresis technique. Large molecule migrates more slowly than smaller ones through the matrix of the gels. The migration depends on the charged particles. Large molecule migrates slowly and smaller molecules migrates highly. This is the separation of the DNA. For the separation, electrophoresis include agarose gel separation. Also, polyacrylamide gel electrophoresis separation. These two techniques are used for the separation of the DNA. Also, agarose is the polysaccharide which is extracted from seaweed. लाकडापासून बनवलं जातं समुद्राच्या. It is used to separate the DNA fragments of 300 to 10,000 base pairs at 0.5 to 2%. Here, this number of base pairs are separated by this agarose techniques. And the agarose from a solid matrix which allow DNA migrate to an electric field based on the size. This agarose is a matrix, solid matrix, which allows the DNA migration process in an electric field based on their size. Large molecule migration is slower and smaller molecule migration is higher. So this slide shows the visualization of the DNA. For the visualization under the ultraviolet rays, one chemical is used. Name of that chemical is ethidium bromide. This chemical is used as a stain. The DNA can be visualized by staining with ethidium bromide. Also, this chemical is toxic. Ethidium bromide is toxic solution. That's why care should be taken during the handling of ethidimer. Otherwise, this is the carcinogenic chemical. Also, this ethidium bromide is an interchelating agent. This is the interchelating karta, which will be insert itself within the basis of the DNA and will exhibit fluorescence under the ultraviolet light. But care should be taken during the handling. Also, single-stranded DNA and RNA will bind with ethidium bromide, but to lesser extent, and will see the structure of the DNA as like a right side, double-stranded DNA. It is stained by metal is ethidium bromide this chemical or dye is used. Then interslated dye places the DNA fragments. We can identify the fragments of DNA. So this slide shows the agarose gel DNA verification. How to verify the DNA. Also, after the separation of DNA, we will get separate the DNA, RNA, etc. How you know RNA? This is the ribosomes. 18 years, 23 years, 28 years. These ribosomes are required for the synthesis of protein. This part is completed in transcription purpose in your 12 standard. Then agarose gel electrophoresis can be used to separate the DNA fragments of different sizes. Different forms of a DNA molecule of the same size can also be separated by agarose gel electrophoresis. Because of its compact size, supercoiled DNA will move faster than relaxed or nicked cellular forms. See this slide. At upper side, application point is there. Lahan खोज this the application point. And after the migration on this agarose, then DNA are separated. First, there is migration is slower of the DNA due to larger size. DNA is bigger than RNA. Then after that, we will see the ribosomes. Ribosome structure is smaller than DNA. That's why migration is higher than DNA. 18 years, 23 years, and 20 years. And some RNA will be degraded. That degraded RNA's movement is faster than DNA and ribosomes. That's why this migration is higher. But these will be diffused on the agarose gel electrophoresis. This will be diffused. That cannot be identified. And this technique is used for the also PCR. After PCR followed by this arm PCR or other PCR used for the detection purpose, fingerprinting purpose, restriction endonuclease purpose, genetic engineering purpose, etc. And here this topic is completed. All of you have the write this demonstration as per your journal at page number 187. You have to write this material as per your journal and draw the diagrams given in your journal. And here this part is completed. I will send this PPT to our group, first year groups. If you will have any problem, read this PPT or call me. Here this topic is completed.